Okay, my good friends, we are getting closer and closer and closer to bringing mud fossils to the world. This is Roger, Mudfossil University, and it looks like we may have the opportunity to present mud fossils in a biological way through a lab which has offered their services. I'm not sure this is going to go down 100%, but no matter what happens, there is a protocol for harvesting these different biological samples that we want to submit. I don't care where you take it. Now, my understanding of what might happen is that if we can come up with an adequate protocol for citizen scientists to document exactly how they harvest what I am going to ask you to harvest, and then they will test it at these labs. Now, this guy's got a ton, he's got like 26 different labs that they test bacterial things for the CDC and all these kind of things. Now, if it happens, I don't know if it's going to happen. He's very, very interested, and I am very interested. So, and he's a realistic guy. He's a guy that wants reality. Now, they have a university, and it's all accredited and all kinds of stuff. It's very upper end stuff. But we need to be certain we're not just making fools of ourselves and submitting things that don't have a good chain of custody. Let's put it that way. So I am going to show you the way to harvest this stuff and explain what the protocols are and what they mean and why we use them. Because if you don't harvest the stuff right, you're just wasting your time. And then you make a damn fool of yourself because you say, "Oh, look what this is," and the guy says, "Yeah, you're a moron." They say that anyway. But at least we can come back and say, "Oh yeah, test it. You test the same." And I have done this right. Oh, they won't test it. But what my friend that has this ability to do this stuff says is that if we overwhelm them with samples, I have 3 DNA tests from my samples. Brian Forrester has DNA tests, at least 1 or more from his stuff he's done. And some of us have done this stuff. Now, mine is very high-end stuff. It was, there was all kinds of solutions and this and that and gene clean protocol kits. And so my 3 tests are extremely well verified. And he was quite impressed with their work as well. Now, what they're going to do in his lab, it appears, is that they are just going to test these things without getting all the deep into the DNA part. There's some sequences of molecules that will come in a certain sequence, like hitting a lottery number. If they are in that sequence, there was life there. So, what we're going to do is present him with all these samples that are taken deep inside of the rocks that we're going to take them from that were originally biological body parts. And there's no question they were. I'll show you the ones I have. I mean, and we have some that are so incredible that, they're new species. Kim has a naga. Tish has, no toes. I have them too. Everybody has these kind of things. But we need to get them put into the mainstream. It's been a very hard thing to get these people to accept this. So we need to create a regular science to this, basically is what it boils down to. And apparently he's willing to maybe even set up a regular university to do this. So it needs to be done. Somebody's going to have to do it. If you leave it up to the mainstream, they're never going to do it, first of all. I can't imagine it happening. It's going to be so crazy. I mean, they got to start over. So we're starting it over. That's all. Just start over and present the stuff and say, this is what we used to say, and here's what we're saying now. That's all. Just get past the fact that they were wrong. They were wrong. Case closed. Now, how are we going to get past this? Mud fossil sample harvest. Strict protocol. Do not you're not, well, first of all, if you don't do it right, you're not going to get authorized. Everything has to be pre-authorized. So you pre-harvest video. You're going to have to do a video showing what you're going to take, what it looks like from a multiple different different angles, and moist, not soaking wet, moist, with full spectrum light. Not that, there's like a 5,000 grade light if you're going to do it inside, which is the full spectrum. If you get those like cool white lights, you can't see the colors that have to be radiated back from the moist biological material, which are the transition metals. That's when we can understand what these things are made of. The red is your ferrous oxide, the O3 style, which has the extra oxygen in it, and it's from the red blood, the artery. That's where we really want to get, because that's fresh blood. Right? You got arterial blood and you got vein blood. What's the difference? Arterial blood is what comes back out of your lungs and your heart after it's been all pumped back up, and all the stuff in your lungs takes out carbonic gases, which were carbonic acids, and it goes through your lungs and it pumps it all up with oxygen from your lungs, and then it just pushes it back through your arterial network, and then it bleeds through and it comes back up through the veins. So you don't really want the vein blood. I mean, I probably would have stuff, but I go for arterial blood. And that's what you really want, because then that stuff is not hard, and it's more of a moist blood. It's like particulates out into like red ochre is what blood is. It's a very powdery sort of stuff once you scrape it up. Anyway, this is you're going to have a pre-harvest video, and then when you do it, of course, you have to be videotaping it, because they want to know. You could spit in there. People blow on there to try to blow the dust off, and they got spit going on there. I mean, a million things can happen. But if you use the right protocols, very unlikely that you're going to have any contamination. So you need gloves, rubber gloves, a mask, drape, which I just lay it around a little bit, to keep everything else off it. Some tape it if you can. A little bleach to clean up the spot before you start. First of all, it's got to be clean as heck. Totally clean. Get all the dust and dirt and everything off. Get it down to and you'll see fabric. You'll actually see fabric. They'll look like stones like this, but if you look at that in a microscope, there's a fabric to this. And there will be a fabric to every single stone that you're going to look at. Now, so you got to get down to there, then you bleach it clean so you get all the worm stuff off, anything that's on there. Clean it good. Right? I don't care how, scrub it good. And then, and bleach your tools, your scraper, your pick, your drills, your brush, everything. You take your brush, what you do, I don't have a brush right here, but just take a rag with a bunch of bleach in it. You rub your brush in there good like that. Get it nice and clean. Let it dry off, and that's when you could, because you're going to have to dust things off as you drill them and clean it and so forth. Don't blow on anything by any means. Anyway, always bleach last before you drill. All right, clean everything up, let it all clean up good, wipe it all down, put your thing down, and then put your bleach on, clean it up good. Then all you need is a gram or two of, first of all, you got to drill. So you're going to drill, you're going to clean the surface, and then in the arterial network, which you should be able to find, you're going to drill down into there just 1/4 of an inch or so, and you get that out there, dust it out with your brush, not blowing on it, and then drill another 1/4 of an inch or so, and get that stuff, put it into your glassine bag. Dust it off onto a clean piece of paper or something first, and then put it in a glassine bag, and then label it what it is, and then put it into another plastic bag. So you insert it into the first bag, and then you seal it that well, obviously, and then you put it in another bag, the second one. Then be certain to video the harvest. As you're doing it, make sure you're videoing it, because nobody's going to believe you. They don't believe a word anybody says, trust me. I don't care how much documentation you have. All right, so, and then all of this, before you can submit it to the lab, I have to go through this, or somebody's going to look at it and say, okay, they did what we asked, it was pre-authorized, they did the video, it looks like they did everything right, I can see they had a mask, they had gloves, it is, then we submit an authorization code, then you just mail it off to the lab, and they will do the tests on it, and they'll send you the results. Right? So that's, and no matter who we submit them to, this is the way you harvest this stuff. And anybody can do this. You don't have to be any special PhD or some hotshot scientist to do this. You just do it this way and you're good. They don't have any special magic. Right? So let's because they're not going to do this. And I don't know why. I don't know why they don't look into this like, wow, look at this. This is fabulous. That's what I expected. When I started this, that's exactly what I expected. And boy, was I wrong. That was 7 years ago. And a lot of money and a lot of time and a lot of frustration, a lot of aggravation. So, I want to get past it, and I want to be considered a normal human being that had an interest in something and progressed to the end of it, instead of just saying, ooh, that's nice, and just walked away, or said, oh, I don't know what to say about that, and I don't want to look like a fool because I've been saying something else. I said a lot of other things. I bought into everything they sold until I started looking for myself, and then I realized, man, were they selling stuff that was just rotten to the core. Everything I'm seeing. And they will not turn around. And they have everybody so brainwashed that they stick right with them. It's like a bunch of little sheep following a Pied Piper. Amazing.